is Yingratana Mclennan, and I'm a PhD candidate in Dr. Martinez Cerdeña's lab, where I study brain degeneration and Fragile X-associated tremor/ataxia syndrome, or FAXTAS for short. FAXTAS was first described in 2001 by Drs. Rondi and Paul Hagerman here at UC Davis. It's a genetic neurodegenerative disease caused by a mutation on the X chromosome, and it leads to problems like tremor, balance, and cognitive decline later in life. So what do I do? Well, first, a bit of background. Here's a coronal section of the human brain. Most research on neurodegenerative diseases focuses on the gray matter, where the neuron cell bodies are located. But what if in some diseases the problem starts elsewhere? FAXTAS is one of those diseases. It appears to primarily affect the brain's white matter, the wiring that connects the different brain regions together. These white matter fibers are actually the axons coming from the neurons, and they look white because they're coated in something called myelin. These fibers rely on different support cells. One is a star-shaped cell called an astrocyte, helps provide the energy, maintain the blood-brain barrier, while another cell, the oligodendrocyte, produces the myelin, protective white coating around those neuronal axons. In FAXTAS, the myelin, the protective coating around the axons, starts to break down. This process is called demyelination. We can actually see these white matter changes on MRI, but we still don't fully understand what's driving them. That's where research comes in. So, to investigate what was going wrong, I looked at brain tissue donated by individuals with FAXTAS. And using techniques like immunohistochemistry and fluorescent microscopy, I could really zoom in and look at all types of brain cells in really great detail. So under the microscope, it was really striking. In the frontal cortex white matter, those normally star-shaped astrocytes, they were breaking down and disappearing. This wasn't just in the frontal cortex too. It was recently reported in other white matter regions. So that's a big problem because astrocytes are essential. They support those white matter fibers, the axons that carry signals from neuron to neuron, and they help maintain the blood-brain barrier. So their loss is devastating. So then the key question becomes, is astrocyte loss actually linked to demyelination, that breakdown of the myelin layer in FAXTAS? To answer this, we ran several analyses, and a clear pattern emerged. The demyelination was most closely linked to the degeneration of those white matter astrocytes. So, as mentioned earlier, FAXTAS is a genetic neurodegenerative disease, so we can actually identify people at risk before clinical symptoms show up. That's powerful because it means we have a chance to step in before brain cells start getting sick. It's possible to support cells when they're stressed or sick, but once the disease progresses and those cells are lost, restoring that system becomes extremely difficult. So moving forward, my goal is to understand why astrocytes are so vulnerable, develop biomarkers to detect when they begin to fail, and ultimately identify therapies that can protect these brain cells before they die off. And hopefully what we learn from FAXTAS can help us better prevent other neurodegenerative diseases. Thank you for listening, and thank you to the ARCS Foundation for supporting scholars like me.